Hello everyone and welcome to Roadmap. So today's topic is plant tissue culture part 2, and if you like the video, don't forget to click on thumbs up button below. You can also subscribe for more video updates, and you can also give your suggestions in the comment section below. So the topics to be covered are introduction, brief history of plant tissue culture, types of tissue culture. In that we'll learn about the callus culture, organ culture, cell culture and protoplast culture. Coming to the introduction, what exactly is plant tissue culture? It is the cultivation of plants from seeds or organs or any part of the plant, and it is done in under aseptic conditions and in nutrient medium, and also explant, which is the any part of the plant is taken, that is root, stem, leaf. And what is totipotency? Totipotency, it is the ability of cells to develop into a whole plant. It is called as totipotency. So already I have covered with the introduction and basic concepts of plant tissue culture in part 1. So what I would request you is that just check the video and learn the concept, so that you will be able to link the video in a better way, and you will have a better conceptual understanding. Coming to the history of tissue culture, about 250 years ago, Henry Louis Duhamel demonstrated callus formation on the decorticated regions of implants, and many botanists regard this work as the foreword for the discovery of plant tissue culture. In 1853, Trickle published pictures of callus formation in plants. A German botanist, Gottlieb Haberlandt, in 1902 regarded as the father of plant tissue culture. So very important, remember that who is the father of plant tissue culture. He first developed the concept of in vitro cell culture, and he was the first to culture isolated and fully differentiated plant cells in a nutrient medium. During 1934 and 1940, three scientists named White, Northcote and Gottfried largely contributed to the developments made in plant tissue culture. Also, good progress and rapid developments occurred after 1940 in plant tissue culture techniques. Stewart and Ray Inert, 1959, first discovered stomatic embryo production in vitro. Maheswari and Guha in 1964 from India were the first to develop anther culture and pollen culture for the production of haploid plants. So this is about the brief history of plant tissue culture. Coming to the types of culture. Now there are different types of plant tissue culture techniques, mainly which is based on the explant which are used. So it is callus culture, organ culture, cell culture and protoplast culture. Now let's learn about it one by one. Coming to the callus culture, this involves the culture of differentiated tissue from explant, which de-differentiates in vitro to form callus. So this is callus tissue culture, which forms the callus from the explant. So it is called as callus tissue culture. Now coming to the organ tissue culture, it is the culture of isolated plant organs. So it is called as organ tissue culture, and the organ used may be embryo, seed, root, endosperm, anther, ovary, ovule, meristem or nucellus. The organ culture is divided into two, which is organized organ culture and unorganized organ culture. So what is organized organ culture? When a well-organized structure of plant is used in culture, it is referred to as organized culture. The characteristic individual organ structure is maintained and progeny is formed which is similar in structure as that of the original organ. Coming to the unorganized organ culture. This involves isolation of cells or tissues of a part of the organ and their culture in vitro. Unorganized culture results in the formation of callus. The callus can be dispersed into aggregates of cells or single cells to give a suspension culture. Now coming to the cell culture. The culture of isolated individual cells obtained from an explant tissue or callus is regarded as cell culture, and these cell culture are carried out in dispension medium and are referred to as suspension culture. So cell suspension cultures.